One of the things that we make all biology students do is to learn about biological specimens by observing them with the microscope and learning to make drawings. And I remember being taught this going on 30 years ago, and I remember grumbling at the time. Even then, we had a camera up on top of the microscope, and we said, why don't we just take a picture of the stupid thing? But the point is that the professor made us draw. And by looking through the microscope and recording what we were looking at on pieces of paper, we learned the anatomy of the structure we were looking at, to the point where I can still remember some of those early drawings today. And the fact that you observe and you try and make a reasonable representation of what you're seeing through the microscope on a piece of paper means that it sticks with you. It helps move the drawing, the anatomy, into your long-term memory. But wait a minute, I'm not an artist. Sure, some people have much more artistic ability than others, and I'm one of those people that doesn't have a whole huge amount of artistic ability, but I can do a reasonable job if I really concentrate. If I look down and I look at the shape of cells, for example, I can record them onto the piece of paper. I tell my students in the practicals that I lead for the organisms that they're drawing that they're not really being marked on their artistic ability. What they're being marked on is a sort of reasonably accurate representation of cell shape and form, and they're being marked on their endeavor, their engagement. Did they try? Did they look through the microscope and really see a shape and try and record it faithfully onto paper? So let's talk a little bit then about how we go about doing this.
What we want to do is try to recreate what we see through the microscope on paper. When you look with the 10 times objective, you can see all of the structures that I want to draw. I want to do the vascular bundle out to the outside of the stem surface, but to do that would require drawing thousands of cells, and as I've said, we're not going to do that. The first thing we might want to do at low magnification, though, is do what I call a plan view. Let's create a plan view. And that is just a really low resolution thing that doesn't include cellular detail at all. It gives some indication of where the different structures are within the piece of the section that we're looking at. And so, for example, if I look along this segment of the stem, the outer layer goes something like this, has a little bit of a jaggy there. And you can see that there are a couple of layers of very orderly cells there. So we could just go like this and draw another line. And there's even a second or third layer that's there as well. So we've got a layer of cells that go like that. Now, if we look further inside, where it gets more interesting, we can see the vascular bundles themselves, and those include the xylem and phloem, the cells that we really want to concentrate on in this lesson. So, for example, there's a big region of cells here, great big cells with red cell walls. Those are going to be xylem. Phloem is in this area here, it looks like. In an adjacent vascular bundle, we've got a great big and even bigger region of xylem conducting cells, and we've got a big region of, let's call them phloem fiber cells. Phloem I haven't talked about yet. It's in between those two structures. And there's even a third region, something like this. You can see that my plan view is not completely 100% accurately representative of what's going on in the slide, but it briefly shows a guide, a signpost to what we want to draw. So, and we want to include maybe some of the bigger cells that are down in this region here. When we go up to our higher magnification view, all I'm going to want to do is draw what's going on in this region here. So I would indicate that on the plan view, something like that. If you're really elaborate with your plan view later on, of course, you can incorporate labels into this. You can describe what these different regions of cells are, what tissues these things are. Now we go from the plan view to the higher magnification view, the actual drawing that we're here to make today. Before I show you the real details of doing that, I'm going to show you a classical mistake that students make. When you look, even with the 40 times objective at the zoomed up view, which I'm showing here now, there are still hundreds and hundreds of cells. People get dispirited really quickly and they start to do this. They start to draw cells. Might be a bigger one over here and a couple of smaller ones in here and something like that. And that's the kind of drawing that you get. My professor said to me, and I will say to you, what's the point of doing that? You're not learning anything. Cells aren't things like this that have discontinuous plasma membranes. They wouldn't be alive. There's no point you spending your time drawing hundreds and hundreds of cells like that, when instead you could take your time and you could look at the structure of the cells here. The big xylem elements like this have pentagon, hexagon, octagon shapes, and what's really beautiful is the way they fit together sometimes. I told you I'm not a great artist. I'm just doing this as representative of what I see through the microscope. Just an example. Now let's go and make an actual drawing.
Okay, so here we go. What I've done on the microscope is I've put the slide up to 10x. And you can start there. You can see the big cells that we want to draw. To draw the smaller cells, you'll probably need to go up to the 40 times objective. But anyway, start at 10. You can see on the page here, I've already drawn a plan view like the one that I showed you before. I've even put the smaller schematic of the entire cross-section in as a guide. You can see the little dotty box here, which suggests the region that I want to show, and you get it. In case I wasn't explicit enough earlier, if I draw this one region, that's representative of the entire population of cells within the root. So we'll go ahead and we'll start drawing what's in here without making the mistakes that I indicated earlier. The easiest way to do this is to pick the very biggest cells, and that's those xylem, the metaxylem conducting cells that we're looking at there. So we'll want to leave some room down in the bottom left corner here for the big cells that are down around them. But what I would suggest is start with the very biggest conducting cell, and it's almost circular. You can do this, put that in there like that, and then try and draw some of the adjoining ones like this. And you can see, again, my artistic skills aren't sort of amazing, but I'm taking my time drawing each cell to start with, and I'm drawing them so that they fit together kind of the way that you see them fitting together there in the actual video. You can see that there are some tiny cells that are down in the corners here like this. So you take your time doing that sort of thing. What I'm going to want to do here, just to speed the video along, is to draw the whole series of cells going towards the outside. So there will be a couple more of these big xylem cells. This one here is almost octagonal if you take a look at it. It goes something like this. Not very good, John, but it'll have to do. And then this one here has a little bit of a wiggle in it like that. And they fit together quite nicely up against this side here. Eventually, I'm going to take my time and I'm going to fill in everything that's in this space here. But for now, we come to a region of little teeny tiny cells. What I'm going to do is just draw some hatches. This isn't the final version. I'll come back and I'll use my eraser and I'll rub that out and I'll put in some of the details of those smaller cells because they're very important. And then we've got a circular region here. I don't know that I've left myself enough room. We're going to cut that circular region off about here. So you see what I mean? I'm even planning as I go along where things are going to go. If you look further to the outside of the cell, sorry, bear with me. I'm just trying to figure out how to move the image around on my computer here. There we go. Okay. There's a 1, 2, 3, 4 biggish cells are going to go here. And then there are those few layers of smaller square cells. So you see what I did? I did a real quick sketchy guide to where all of the different cell types are going to be. Now what I'll do is I'll start drawing them and I'll bite my tongue and I'll take my time and I'll sweat over it and I'll make a drawing that's as good as I can get. But instead of occupying all of your time, what I'll do is I'll just make sort of time-lapse images as I go along while I'm doing the drawing. So wish me luck. Let's see what happens. Okay, well, there you go. That took me about 20 minutes. And it's no Michelangelo, let's say, but it's not bad. I mean, I would give it very good marks. For me, I'm quite proud of myself. You can see that there There are little circles in there because there are little tiny cells in the image. If you look at them carefully, I've indicated thicker cell walls in some and thinner cell walls in others. It's just a continuity which shows all of the different cell types. And I've indicated the labels with the word label here because I don't want to give it away. I want you guys to go figure out what the labels are. So there's a short, maybe even somewhat longer than I want it to be, lesson in how to draw. As I say, 20 minutes for me to do that. If you take half an hour to draw the one stem and then half an hour to draw the other stem, that'll be pretty good, I think. Okay, carry on. Good luck.